everyone and to everyone online. So this morning, I want to talk to you a little bit about artificial intelligence and how it can be used in biology. And we'll talk a little bit about sort of challenges and where are we going next. Interesting. Okay. So before we get started, I want us to all get on the same page a little bit, right? Because terms associated with AI, ML get tossed out all the time and are often used synonymously. And so I think that's okay to use them synonymously, but let's talk about what they technically are and what they actually kind of mean. So AI, or artificial intelligence, right? This is a very general field that's kind of all encompassing. And it really involves trying to simulate how humans use their intelligence, but you're doing this via machines and programs, right? So we think about, how do we learn? How do we solve problems? How do we make decisions? But it is sort of like this big umbrella. And within artificial intelligence, then we have the field of maybe. I do not have the ability to. Oh, okay, thank you. So within the field of AI, then we have machine learning, right? Machine learning is really focused on, we talked about doing these human processes. Machine learning is really focused on finding patterns in decision making, and then making predictions based on those decisions. So if we just look at what we have here, right, perhaps we have data where we have, let's say, proteins that are connected, and we know types of some of those proteins, either they're red or green proteins in this case. We might be able to use machine learning to find patterns in the data and then make predictions about unknown proteins. Within machine learning then is deep learning. And really the goal of deep learning is very similar to machine learning, right? We're still looking for patterns in the data. We're still looking to make predictions. But instead of using more what I'll call traditionally statistics-focused algorithms, deep learning comes with these what are called neural nets. And basically you have an input layer of data or features, and those get reduced and transformed sequentially until you get to an output layer to make a prediction. We'll talk more about some of this later too. Within deep learning then is the field of generative AI. Generative AI, I think, is where AI and these subfields get confused or get used interchangeably. Generative AI is really about, can I take some type of content? That could be an image, it could be a video, it could be text. And can I learn how to create those things from existing examples? I'm almost done. So within generative AI, then we have large language models. And I think LLMs are the thing that most people are familiar with in the recent last 3 years, right? The only thing that distinguishes them from generative AI is they're explicitly trained on text or language data, and they're used to understand, generate, and to manipulate the human language. And within that, then, one example of a large language model is ChatGPT, right, which I think most folks are familiar with. So as you hear some of these terms in the coming week, it's important to remember that a lot of these terms get used synonymously, but they all sort of are born with different types of methods and goals, and they're born out of certain types of requirements too, in terms of computational power and architecture. All right, so I'm hoping you'll allow me to stand on my 2-minute soapbox and tell you that AI did not just happen 3 years ago when ChatGPT existed or got made, right? The concept of AI and actual applications go back to the 1950s, and actually probably before that too. So my soapbox is that all of the things that exist today are built off of mathematical and statistical foundations of the people who did these things on pen and paper and chalkboards. So when we think about artificial intelligence, I am going to go through just a very brief timeline that's very incomplete, but to give you a little bit of an idea. We talk about artificial intelligence that started in the 1950s. And the timeline that I have here is really where things kind of started gaining significant traction or popularity. So machine learning in the 1980s-ish really became popular, right? And deep learning started gaining traction in the 2010s, and generative AI in the last 3 years. Okay, and I want to talk about a little bit of why that is. So I mentioned I'm going to give you the briefest of brief histories of AI and hope that that helps a little bit. But also note there's going to be a lot of holes in this because we could be here all day if that's the thing that you wanted me to talk about, right? So AI was technically born in 1956. Some actually would argue it was born before that when Alan Turing did the Enigma, if you've seen that movie. But most people recognize that AI started in 1956 with these three guys, Newell, Simon, and Shaw. They developed what was called the logic theorist. Right? And that was the first tool that was designed to perform automated reasoning. And in the most exciting of application areas that you could think of, that tool was designed to solve mathematical proofs on its own. Couldn't have thought of a better solution and application area that would be useful worldwide, right? But to them, this was really exciting. And what they ended up showing is that these programs could actually solve these mathematical theorems in an automated manner and could do it more efficiently than humans. So they organized and held a conference and coined the term artificial intelligence. And like all good science, it was met with a pretty lukewarm reception, with a bunch of critics saying, like, I don't know how useful this actually is going to be or how impactful to the field it might be. So going down that timeline, right? Machine learning was technically born in just a few years later in 1959. Arthur Samuel, who's pictured here, is the first person to coin the term machine learning. He developed a program that calculated the probability of winning a checkers game. So you might ask, why is this a machine learning algorithm? Because, at least as a statistician, I would tell you, at any given point in a checkers game, you could enumerate all the possibilities of how this checkers game would go, right? In theory, if you had all the time to sit down. So you don't need machine learning because just calculate all the possibilities. The problem is there are so many possibilities to enumerate. And back in those days, the computational power to enumerate all those possibilities didn't exist. So instead, what he did was he developed an algorithm and a model that mimicked a lot of these possibilities, but didn't enumerate all of them. He fit this model and then started generating probabilities of winning a checkers game. I sort of like to think of it as, I don't know if any of you have ever watched poker on ESPN. Every time they get a hand, it will put the probability that somebody's going to win that poker hand, right? Maybe back in the day, I like to think they had announcers around checkers boards talking about probabilities, which I'm sure is not the case. But machine learning really gained traction in the 1980s, 1990s, because computers got better. And the field really decided, instead of trying to do everything that we're talking about with AI and doing human reasoning, they decided to really focus on solvable and practical data-driven problems. So finding patterns in your data, being able to model those patterns and make predictions. All right, out of machine learning then comes deep learning. The first instance of deep learning actually happened about 10 years later then. And what was developed was called the group method of data handling. Essentially what that was a really fancy way of taking something like polynomial regression and automatically generating what are called neural nets or deep neural nets. They are kind of what they sound like. You have different nodes that are pieces of data, and they're designed to connect to one another, sort of like the neurons in your brain are thought to connect to. And what happens at each level of a deep learning model is a bunch of really black box data transformations of your variables, right? But when you start to understand how to develop these layers, pretty powerful modeling capabilities come out of this. The term deep learning, I put this in here because it's about the only place you can see it in this history. The term deep learning was coined by Rena Dechter, who is a female. in the field, and one of the very few that I could find in sort of the substantial progress and history of this. So I thought it was really cool. She's a professor, and if you want to read more about her, I thought she was really interesting. But deep learning really took hold in 2012. There's a competition called ImageNet, where they try to do image recognition tasks, right? So if I show you a picture of a dog, you can tell me, hey, that's a dog. Well, if I show a computer a picture of a dog, can it tell me, hey, that's a dog? And this model called AlexNet in 2012 won this competition using deep learning. And that sort of caused the field to actually really gain some traction. That along with, right, we have different types of computing power. So we have CPUs and we have what are called GPUs. GPUs started becoming a household thing then. I mean, expensive, but they were available. And those are the types of computational systems that allow us to train these really deep neural nets. All right. Last one here. So if we go one more deep down into the level, we have generative AI. Right? Generative AI was actually born in 1906. The only people who knew about it are mathematicians, because I'm sure nobody else cared at the time. Nobody ever cares about the math majors. No. So in 1906, the first manuscript on Markov chains came out, and Markov chains are known to be able to process text data. Right, but where we started to really see movement in this field is in 1972. Harold Cohen, who's shown here, he developed what was called the Aaron computer program. He was an artist, actually, by trade to start with, and then developed a computer program. This program produced paintings and drawings in an autonomous way. And then, as most of you are aware of ChatGPT, right, in 2022, OpenAI launched ChatGPT, and this field has taken off since. Pretty much every day, there's a new company out there using this. One thing I wanted to just briefly mention in all of this history, right? Where does all this growth in popularity and usage come from, besides the fact that the things that were done turned out to be useful? Well, there is a very direct link to milestones in computational power, right? Their improvement in that power and their availability to the general public and research community. We also have an increased ability to store and generate data. So if you think about the amount of data that Google, Amazon, all of these companies store, as well as if you look over at EMSL, right, the amount of data and samples that they can run now is significantly greater than it was 10 years ago. Additionally, we have things like open source programming languages and code frameworks. So I can go grab Python. I can actually go grab a link to open source large language models to implement my own research. Right? I could do that without a cost. And for users who don't want to develop models but just want to use them, right? We have ChatGPT, we have all of these engines where you don't actually have to know how to code. So accessibility and computational power, I think, are the primary things that drove popularity of these things. All right. Where I'd like to go next is I'd like to just show you a few applications. These by no means are, right, this is not going to be comprehensive in any manner. There's no way to do that. I just wanted to talk about some cool applications in the field. And the first one I want to talk about does not belong to me or anyone at PNNL, right? But some of you may have heard of AlphaFold or AlphaFold 2. So what this is a deep learning model that is used to predict protein structure. Right, and it predicts protein structure based on just an amino acid sequence. It goes and computes how close is that to other amino acid sequences in the database, finds the ones that are closest, and then uses a database that the model's trained from the protein data bank. And what I want to note here is that there are over 215,000 entries of structure of proteins in that database that were all experimentally determined and validated. So AlphaFold stands on the shoulders of manual research, and a lot of it. But it turns out it's a pretty cool tool, and it also as the statistician or one of the statisticians in the room, it will also provide to you confidence metrics of their predictions, which is an important thing to pay attention to. Okay. I want to show just a couple of examples of some of the research that we've done here at PNL that involves AI. So one piece of this is where we have used machine learning to try to find biomarkers. In this case, I want to talk about biomarkers for type 1 diabetes. Right, so on the left, you have the image, you have a healthy system. And at the bottom, in the case of type 1 diabetes, right, we produce less insulin. And so then being able to manage things like glucose becomes really difficult. Okay, the trajectory of type 1 diabetes, just very briefly for you, right? The TEDdy study, which TEDdy stands for the Environmental Determinants of Diabetes in the Young, so it's childhood type 1 diabetes. What happens is a child will be born into one of these hospitals around the world where they have funded this research. They're subjected to a blood test. That blood test comes back with an HLA marker, and the doctor says to a parent, hey, your child has a genetic predisposition to type 1 diabetes. That genetic predisposition is pretty high. We're talking like 90% of these kids actually will develop type 1 diabetes based on this blood test. And they enroll them in this study and follow them every 3 months for up until their teens in a lot of cases. So a huge cohort of people. The progression of the disease itself, right, is we've identified these kids with genetic predisposition, and some combination of genetic and environmental factors then starts to take normal glucose and sink it into the abnormal range. And we have tests to determine when this is happening through autoantibodies. So as a child starts to progress towards type 1 diabetes, they become what's called autoantibody positive. And eventually, when things really give out, then they're formally diagnosed with type 1 diabetes, and that usually coincides with symptoms of diabetes. Okay, so PNL is involved in this large study. There was a discovery phase and a validation phase, and I'm just going to talk about the validation phase. So this study actually uses case control partners with samples. They're partnered based on things like age, which clinical center throughout the world that they came from, sex, and the controls are still kids who are genetically at risk as well, but just at that age have yet to develop either autoantibody positive or type 1 diabetes. So what was generated at PNL was thousands and thousands of samples of targeted proteomics from plasma, right, where 811 peptides were measured. We have these case-controlled match samples with two outcomes, either autoantibody positive or type 1 diabetic. And we wanted to evaluate the ability to predict, could we predict what your disease state will be, but could we do that at some time point prior to the actual development? So what I'm showing results for here is kids that are at a time point 6 months prior to developing type 1 diabetes or autoantibody positive. What we did was it's a very basic statistical model is logistic regression. Right, so my outcome is yes, no, or control and disease. And I can fit that to my data with all 811 of those peptides, but I can also do what's called a lasso penalty that I throw on top of that does automated variable selection. So one thing you'll find in the field of machine learning, not deep learning, but machine learning, is that often you have so many variables that you're trying to use to make predictions, you need some way of selecting or downweighting some variables so that you're not trying to use all of them. And that's the approach that we took here. So I do have the publication information there for you. When we fit these models, right, what I'm showing here is area under the curve. 0 would be the worst in that case. 0.5 would mean you might as well flip a coin. 1 means you can make perfect predictions. But just off of some proteomic biomarkers, we get AUCs up by 0.9, which is really good for real-world human data. And what we can also get out of that is then because this lasso model does variable selection, right, we can say, well, which proteins were actually used in making these predictions and how important were they? So what we can just at a very brief glance see is that a lot of the markers that came up for autoantibody positive are not found in the type 1 diabetes model, but most of the markers in the type 1 diabetes model are found in the autoantibody positive model, but it's a subset. So this gives researchers a place to start and think about, can we develop assays for these diseases? Presumably, in the case of autoantibody positive, at least, right? If we can get ahead of that and think about interventions and a way to slow down the progression to type 1 diabetes. Okay. The next application I want to talk about is more machine learning, but for spatial biology. And I should give credit where credit is due. So David Degnan, who's in the room, is a part of this research as well, right? Hopefully most of you have heard of spatial biology, but essentially what it says, right, is if I'm looking at tissues of living organisms, their spatial location and the context of where they live next to other cells might matter in the function and the role that they have in the biological processes at hand. Right? And this has become increasingly popular rather than doing what we did in the Teddy study where you have a plasma sample and you just say, let me take all the cells, all the information together and see if I can parse out information. So what researchers will do is they will take what's called an optical image, which is what I have on the left. This is from a study involving placentas, right? So I have the information for it there, but they'll take an optical image and it's maybe a little bit hard to see, but we have this zoomed in region. They have different types of cells in the placenta that are called STB or core, and that was the focus of this work. In addition to that optical image, then depending on whether you want proteomics, transcriptomics, in this case, they did metabolomics and lipidomics, right? You can start to get maps of each metabolite or lipid as it pertains to that tissue. And then, for example, the region that we have zoomed out here, right, we can start to ask, hey, do I start to see intensities that look really similar in that region of one of those features that I don't necessarily see in other places in the image? In that case, we might be able to start to say this metabolite or lipid is playing a role in whatever process that we're interested in. All right, because this is more focused on the computation aspect of things, what I want to tell you is when scientists do this currently, they say, I want to find biomarkers associated with whatever, disease X, process Y, right? They will look at an image like this and say, hey, there's different regions or different types of cells that I'm interested in. And then, sadly, by hand, they will identify and annotate all of those regions, which is super manual and time-consuming and sad, because scientists who are this smart should be spending their time doing other things. So in the field, there has been some work, and what we tried to do was look at, can we make this process easier? So one thing I want to tell you is, if you're trying to do something, you know, Google or Amazon or somebody else probably has already, quote unquote, solved the problem. So in this case, we have Google's segment anything. You can drop a photo in there and it does its thing right here where it separates all the different types of objects in the picture. And it does it seamlessly, it does it within seconds. It's really impressive. Okay. This model was trained on over 11 million images, just for context. So you aren't going to build this in your own backyard. And when you take an image like we're interested in on the top, where it's manually annotated into regions, I drop this into segment anything, and it couldn't determine that there were any difference in regions at all. So not surprisingly, Google's not super interested in mass spec imaging. And this made David really sad, but happy, I guess, right, David? Because that meant that you had work to do. All right, so the approach then we take is we said, like, okay, are there viable algorithms where we can identify these regions of interest in an automated manner? In machine learning, I know that Javi's going to talk about this as well, but there's two concepts. There's a concept of supervised and unsupervised machine learning. Supervised machine learning means somebody went in and manually annotated a bunch of data, right? They said, in the case of this image, they went in and did what we've shown here. In the case of other things, they've gone through and they say, I have a proteomic profile and I know this person is diabetic and I know this person is not. And then we try to learn and train a model based on those annotated things. So there are tools out there that exist where it's been based on supervised learning. What I'll tell you is it takes a lot of data, and a lot of time that data is really specific to the types of imaging that you're doing. So we set out and said, well, can I look at unsupervised machine learning? And what I'll show you is on the left here, or on the right, sorry, that is one of the original images that we used. It actually was broken down into smaller tiles and we stitched it together. And we had our collaborators here at P&L go in and manually annotate some of those regions, which is why you're only seeing some green and blue and yellow. But we're going to use those manually annotated regions to try to see how well does the machine learning algorithm actually match what an expert says. Okay, we considered these types of algorithms. Kelly, what time do I have until? Okay. So just really briefly, the top three are called thresholding. It's exactly what it sounds like. I look at my image. If I'm looking at a black and white image, it's looking at, how light or dark is the pixel of the image. And at some threshold, I say everything with an intensity greater than this belongs to one group, everything lower than this belongs to another group. You can do this for more than two groups or types of regions. We also have statistics-based clustering methods, which again, I'm pretty confident Javi's going to talk about some of these clustering methods. But basically taking characteristics of those pixels in the image, right, and trying to cluster things that look more similar to one another together. There are a couple of methods which are called superpixels. If you think about an image, if I take one pixel of it and I take some pixels that are around that pixel and start to make a superpixel, right? And then try to figure out what that superpixel belongs to. And then we have PyTorch tip, which is a traditional machine learning method. So it's sort of models get more complex from top to bottom. And if we take a look at that original image in the manual annotation, this is what we get out in terms of segmenting the image. So what I'll point out is some of these methods look like they actually do pretty well. If we just take a look at, say, k-means or Clara, you can see that at the top there, yes, no laser pointer, sorry. At the top there, some of those regions where you have pretty distinct areas are popping out. You look at image F, that was one of those superpixel regions or methods, and it looks like it took those superpixels and maybe made two course of choices. So we did this on this image. We have actual metrics of accuracy that we investigated, but for the purpose of this talk, we won't go into all of that. I will say we also did this on the image at the top here, where the target we had is actually a pretty simple way of determining regions. We have two regions and background, and one of the regions is just that orange dot. In this case, we see some interestingness in results, right? K-means, which did really well before, doesn't look awful, but some of the other methods that looked pretty good before are not looking so great now, like recolorize. And what I'll say is, so image G was the one that I pointed out on the last slide where I said, oh, that's a little bit too coarse. In this case, it probably looks the best or the second best with binning out of all of them. So the only point that I want to make here in terms of this is the algorithm matters, and it matters in what you're actually applying it to. So the next step here, if we really wanted to be very AI about this, would be, could you autonomously decide which of these methods is going to be the best without having to go through by hand? Okay. Last application area then for me. So AI and machine learning for network modeling. All right, so network modeling in the biological systems is pretty common. In this case, I'm going to talk about, let's say that you're measuring proteins. A lot of times what researchers will do will be they gather that data, they'll look at intensities of different proteins across samples, and they'll say which proteins look most similar to which other proteins, and they'll do it using a metric like simple correlation. Right? And then we try to take a look at the network that we made and make some sort of biological inference. Or in some cases, you might make one network for a control condition, one network for an experimental group, and say like, well, what's different about these networks? Which in this case is really awesome. You can totally do that. When you measure thousands of proteins, it starts to look like hairballs. All right, so as part of an internal initiative here at PNNL, we proposed to do this network modeling, but we proposed to do this in a Bayesian manner. And all a Bayesian manner means is that we said, in addition to computing and estimating these networks, we're going to allow, can I take what I already think I know about the field and research in the field and use that as my starting place for my model? So we propose, hey, what if we were able to look through literature where people have studied protein interactions for decades? And what if I was able to pull that information, because I'm not going to sit down with a scientist and ask them to tell me all the things that they know. That actually is a thing in Bayesian statistics. If you're modeling simple things, you sit down with an expert and you say, hey, what do you think the probability of this is? That person's going to hate you if you're doing that with 2,000 some proteins and all the pairs of them. So can we do this in an automated manner? And then fitting these two networks and using machine learning to say, what's the difference between these? So for today, I'm just going to talk about that prior knowledge piece. All right. Again, David and others, right, involved in this project. So we built a pipeline where a user could come and say, hey, I'm interested in E. coli and metabolism, and I only want to look at publications since 1980, where they can pull all of the papers from multiple different databases, take out duplicates, then pull the full text and or abstracts where full texts aren't available because of paywalls, right? And now we've got a bunch of text data, which hopefully from the beginning of this, if you remember, hey, I've got a bunch of text data, that probably means I'm looking at using some large language models or some generative AI. Okay, before we can go and try to use tools like that, trust me when I say this is not where you wanted to start using large language models. It is a mess otherwise. So what we do is we'll take sentences, use a machine learning model called Spacey, identify which of the things in the sentence are biological terms, and then we can use a large language model like Llama to label what type of a biological term that is. As long as I tell it, these are the types of biological objects I'm interested in. Can you please label them? Okay, then we take all of these sentences that have at least two terms in them, because if you remember, I'm trying to find out which proteins or which biomolecules are related to one another. As long as they have two terms in there, then I'm going to use text processing to try to say, hey, according to the text, are these things related? Because we're talking about thousands of papers with thousands of sentences each. Okay. So text processing, also not a new field. There are things that we'll call classical algorithms. Those are things like really simple things like sentence co-occurrence. So you ask, do these two terms occur in the same sentence? That algorithm is going to say, yep, they do. So we're going to call them related. We have machine learning algorithms like PubMed, BERT, right? So those are machine learning methods that are specifically trained on biological data or biological text. And we also evaluated 3 large language models. And if you want to know the full results and methods of this, we've got the paper up here for you all. One thing I want to point out with large language models. So they are great. They can do really powerful things. We all know they have limitations. One of the more frustrating things, I think, in this process, we could talk to David afterwards and see if I'm wrong about this, but is I can ask it and say, hey, here's an abstract where I know these 2 genes are said to be related. And you ask one of these large language models, hey, the interaction between these 2 genes either exists or it doesn't exist based on the text that I gave you. Does a relationship exist? Yes or no? And a large language model will come back and say, hey, first you should measure this gene, blah, 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 blah. Okay, so sometimes you have to actually reinforce train these large language models and say like, hey, by the way, ask for a yes/no answer. That's not what you gave me. Please give me a yes/no answer. And the reason that I bring this up is, right, David's not typing all of these queries into ChatGPT or something like that. We're doing this in a very programmatic sense, right, where you're saying like, feed it one sentence, feed it another sentence. And if you can't look at the results of each sentence coming out, you don't know if you're getting back what you really wanted. So we are still not at sort of the autonomous automated part of this right now. Context matters to large language models. Somebody on a project team I had just asked it to generate a joke the other day that I thought was awful because it doesn't in my opinion, doesn't understand the intricacies of humor, right? So we all have limitations still using these methods. These are a lot of metrics we use. So we took E. coli, which is a pretty well-studied organism. We used information from actual biological databases as to which proteins were related to one another and said, okay, these are the things that we should find based on manuscripts that these models should say are related to one another. So at the end of the day, what you do get back from each method then, after each pair of proteins is evaluated, you get back one of those hairballs for each method, right? And here we're just looking at a bunch of different types of metrics associated with that final network. The red line represents what we should have gotten. And I think the thing that you'll notice is the methods that are blue, so those are machine learning methods, but not large language models. They tend to kind of do their own thing compared to those more classical text processing methods or the large language models themselves. And if we just take a look at some example networks, right? On the left is what we should have gotten. These string methods or basic methods, you can see those look really, really, we have a huge number of connections compared to what we should have gotten. The large language models also look like maybe they're saying too many things are related, but on a much lower scale. And actually, the simple neural net machine learning models look like they do okay. What we found is if you really want to make sure you're not saying that 2 things are related and getting a false positive, probably the neural nets are your best bet, right? The cost of that is they're also not going to say that many things that should be related are related. So it depends on what you're using this for. In our case, we're using it for prior information. If you know anything about Bayesian statistics, that information does not have to be perfect. It is just a place to start. So a large language model in that case might actually be your best bet. Okay. Sorry, that was a lot all at once. But I want to just briefly talk about, so where are things going? What sorts of challenges do we need to take into, and what are things we need to actually consider? All right, to me, one of the big areas that AI really stands to make an impact is on autonomous experimentation. And good news for you all, you're going to hear a lot more about this on Friday. All right, but we have things like where we can take lab workflows and try to automate a lot of them. So that means, actually designing things, executing lab protocol. That largely needs to be done by integrating robotics, right? But the idea would be then, instead of being subject to when a human could go into the lab and do all the things that you need to do, robots in theory can run 24/7. And a part of that too is being able to adapt your experiments. So can I adjust experimental parameters if it looks like things aren't going right, if I'm not getting the result that I don't want? All right, some challenges with this is every system that you're studying probably isn't going to generalize to every other system very well. Right? And it probably is going to cost a decent amount of money to buy all of the equipment and robotics and things like that to make this necessary. The other thing I'll say is, hopefully you picked up on what I was talking about today and say, like, actually a lot of this required a lot of data. And in biology, we don't always have a lot of data. We could, if we could figure out how to start to leverage data together from other labs, right? But that's its own problem. And the last question then you have to make is, so I'm going to generate all of this data. Some of it can probably be analyzed on the fly. But at the very end of the day, if I want to start to make biological inference and conduct statistical tests, that's going to depend on the application and probably depend on some expert's domain knowledge. How do you scale that up if you can scale all the data generation up? You said 25? 35, okay. I sweat for a second there. All right. The other piece where this, in theory, right, I talked about at the very beginning, the goal of AI is to think like a human. And the piece that's really missing in all of this is if you all go do an experiment, you're going to find some results, analyze them, and probably think about like, what's the next experiment I should do? That is not something that is widely done right now using AI, but definitely is a place that we can think about things going. Right? And the way that we can do that is using these algorithms to find patterns in the data and perhaps find biomarkers that we didn't know existed before. Do things like modeling biological processes, right? So things like we're talking about protein structure, or there are models for protein folding. And this, what I'm calling is the fusion of heterogeneous data sources. And so I'm thinking something along the lines of what you saw an example of from me here is, hey, I could use deep learning or a large language model to analyze text data and put that in a format where it can be used. And maybe I could take my mass spectrometry data and put that in a format where it can be used and try to get those into a compatible format, right? Getting that into a compatible format oftentimes requires a lot of transformation, and that's something that AI or deep learning models can help with. Then your next problem becomes, how do I interpret any results that come out of that? But again, the primary challenges here are, do I have enough data available? One thing I talked about, right, is enough data. We could go use data from different labs and reuse data. And if you've ever tried to do that, good luck. Metadata is never stored in the same way. Data format is never stored in the same way, right? So having things be consistent, or at least being able to get them easily into a consistent form, is definitely a challenge. There's a reason that NIH and other agencies have calls out for AI ML ready data. Because when you look at the swath and plethora of data out there, it's great. There's tons of it, and none of it currently is easily usable when you want to combine things together. One other thing I'll point out is when we were doing this example of let's look at manuscripts, right? And if I want to generate hypotheses, you also, I mean, this is a problem in science in general, but you do your experiment and you find out, the hypothesis I had wasn't correct. And then you don't go publish a paper about that. And you leave it for somebody else to find out the same thing, right? You as a scientist can note that and say, all right, my next experiment isn't going to have to do with this. But if there's nothing published on the fact that this didn't work, well, I mean, somebody could go do the same experiment. That's an issue. Also, if I want to use things like a large language model to say, hey, what things are related and what things aren't, I actually can't really ask what things aren't because we're not publishing on that. All right, at a very high level, some other things that you could think about, right, are, I see out in the audience several of you wearing an Apple Watch or a Fitbit, right? So could we use those things and think about them being hooked up to somewhere where that data could be analyzed in a live manner? Could we do biodiversity monitoring using drones and analyzing that data? Things in personalized medicine, particularly because we could start to do things like gene editing and things like that much more easily, or organism engineering. All areas for growth. There's a ton more that I didn't list here. Okay, I'm going to stand on my last soapbox now and just say, what are some general things that you should think about? I've already kind of harped on this. So data quantity, quality, and availability are really, really, really important. Some of that is data formatting and metadata. Is that ready to actually be fed into some of these models? The other thing we haven't talked a lot about is data privacy and security. I don't know if you've ever had this experience, but I just did recently. Our insurance company called me and they're like, hey, we set up this new service where you get a nurse that you can have available to you via email, via text message, everything. We're going to go through your entire medical record with you now. And then you're like, oh yeah, they know everything about me. Well, if we start thinking about using wearables to detect disease, we're going to need a lot of training data, which is going to be data about people, right? How do we keep that type of data safe and secure is a big question, especially when you're feeding it into things like large language models where we haven't really determined how to ensure security and safety there. Google it, you'll find tons of articles. The other thing I want to point out is all of these new techniques, especially in generative AI, they cost a lot of money, right? And additionally, just to throw out there, they cost a lot to the energy and environment as well. So training a large language model is something you will never be able to do unless you become a millionaire, billionaire, right? The companies that can do that are OpenAI, Google, Microsoft. And every time they train one of these models with billions and tens of billions of parameters, hundreds of billions of parameters, right? It's estimated that it costs 1,300 megawatt hours, which basically equates to the annual electricity of 130 homes in the US. So every time we're making one of these, you're using a lot of energy. And if we're not thinking about how we can cleanly and safely generate more energy, it could become a problem. Bias is also an issue to think about. So large language models, machine learning models, deep learning models, all of them, they're only as good as the data they've been trained on. And they are very biased to the things you've shown them because they don't know about all the other things that they haven't seen. Right? And if you've ever used a large language model, they really like to try to make you happy. They really like to tell you what you want to hear. So there's some inherent bias in those types of models. In all of these fields, being able to interpret things is a challenge, right? There's a reason if you look at these types of models have gained far more traction in real world applications than they have in science, because as scientists, we need to be able to explain why something is. Or if I say I can predict who's going to get breast cancer, that's great, but I don't want to, I don't want to go collect a genetic panel, a proteomics panel, all these things from a person. I need to be able to build a pretty simple assay. And my last piece of the soapbox then is just the ability to develop these models like generative AI is limited to a few companies, right? So these models are also going to be biased to the things those companies are interested in. All right, I've done a lot of talking. The last thing I want to say is, I think Moses said at the beginning, right? You want to think critically about these, and I'll go back to probably my favorite quote from a statistician, which is, all models are wrong, but some are useful, right? And where the models are useful depends on the type of data and the type of problem that you're solving. And so I would like to double down on what Moses said, just encourage you throughout your week here to really think critically. With that, I am done, but thank you all. I'm sure there are no questions, right, Kelly? I think they have a mic for you.
Hi, nice presentation. I really enjoy it. And I have a question about like some of the language machine learning tools.
Mm-hmm.
Is it possible that maybe they're working fine, but if the confirmation input from the people that use it can make it work bad? Like I don't know if it has happened with ChatGPT, but maybe it's doing good predictions. But since everybody can use it, they can just say like, ah, yes, yes, you're correct, you're correct, you're correct, with things that are not correct, and they can.
Yeah, it
Thank you.
definitely is a concern. Even within one person, I think I saw an article in the New York Times or somewhere where they talked about like people with very extreme political slants, how it started leaning towards them and confirming their beliefs instead of being an objective observer. So I did actually try last night to tell ChatGPT that 2 + 2 + 2 was 9, and it never caved. But I think when you start doing more complex things, and as they start to retrain their models on the things that are generated in there, you run a real risk of that.
Hi, nice presentation. I have a question regarding how well these models are trained at handling batch effects. For example, if I'm doing a meta-analysis and I have data generated from different machines, say sequencing platforms, or even in case of proteomics, maybe different brands would have a different way of generating data. So how good are these models at handling that difference?
I think the short answer to that is they are not great at handling batch effects. One way you can handle batch effects, right, would be to give it information about which batch each sample comes from. Now, the only problem with that is, let's say I want to train a model to make predictions, and I give it the batch information so it can account for that. Now, if I want to make a prediction on new data that comes from a batch that was not fed into the model, I actually can't make that prediction. So it's a really valid question, especially when we're in the field of biology, is methods to handle batch effects need to become better, need to continue to be developed, because they aren't interoperable. And if your ultimate goal is really to make predictions, it's going to be prohibitive.
Thank you. So I think a lot of one of the big issues that comes from like experimental data is like a lot of it's not very like clean as in like resolution, especially when you get in like the micro scale. Are there models, or how efficient are some models at maybe like cleaning up or segmenting data, kind of like the Google segmentation, or like clarifying resolution?
Yeah, that's a really good question. I don't think I have a satisfactory answer off the cuff to give you, but can try to answer that in the Slack channel for you.
For sure. Thank you.
Thanks.
Thank you for the wonderful presentation. That's so great. I just have some questions about the last clause, which is like all models are wrongs, but some are useful. I'm just wondering, like, as a beginner, if you want to check if a model will be very useful to you, how do you go about it? Because we know that one of the things we always talk about is like benchmarking. Like, you want to benchmark our models against maybe something that is already established or something like that. So is it about being better than the benchmark model, or you just found that these come from a very good data quality or something like that? What should we look for basically?
Yeah, so I think there are a couple of things, right? One is if you know that a model has been shown and well established in the field to be useful for certain types of data, right? So the example I'll give you is transcriptomics data. If we're modeling that, there's been a lot of research that shows I can fit a zero inflated negative binomial model like EdgeR or DEC2, right? And that's known to work pretty well on the data. The other thing we do if we're in the ML AI context is we'll split our data out into a big set of training data and then testing data. And we'll look and say like, hey, how well does this model work when I fit it on this data and test it on something I've never seen before, right? Which is super useful and helps you. Oftentimes, this is how practitioners choose which machine learning model to use. My question back to you then is, or anyone, is, okay, great. How do I do that in an autonomous way if I want to use this for AI, right? How do I evaluate that? And the best answer I think I have is those types of processes are going to have to think about the types of data, the types of experiments. You know, if I had come in and I say I want to study E. coli, and I pull all E. coli data, like, well, is it okay to pull this other organism too to get more data? And if I have enough data, then maybe I have enough data to do that internal sort of model selection. But how to do that needs to be thought about really carefully.